All right, let's start our last section. This is it. We've only got about 3, maybe 4 days worth of material left. We are all done for this school year with content. All right, so this last section is about something called cladistics. So we spent the previous section talking about classification and how we put organisms into different groups. Cladistics is the method of classifying organisms. So this is how they do it. Method of classifying organisms into groups of species and these groups of species are called clades. So now a group can be any sort of big group. I mean, a group could be a kingdom, a phylum, a class. I mean, it could be any of those things. And so we have a name for these groups, and that name is clades. So each clade isn't just like one common group of organisms, but it also consists of their ancestors as well. So it's ancestral organisms and their evolutionary descendants. So all of the things that have come after them as well. So the pictures that we use to show clades and ancestral species are called cladograms. And I've got a couple examples of cladograms right up here. You might see some of them that are sort of organized in an angular way here. You might see some of them that are organized in kind of a straight up and down kind of way. They both show the same thing. But on a cladogram, clades include, like we just said down here, ancestral organisms and their evolutionary descendants. So these blue and green examples over here are examples of clades because we can see this ancestral organism that they're coming from down here. And notice how this thing doesn't continue up. This line doesn't continue up, and that's because that ancestral organism is extinct. And this then therefore includes all of the descendants that came from this one ancestral organism right here. But this blue one is also a clade because it includes this ancestor plus these new species here. This is not a clade right here because this clade does not include this group of organisms over here, which are the descendants of this organism right here. And so this can't be a clade. It includes this over here. If you wanted to include all of these here, this clade would need to be here. You would need to be highlighting this here. And the same goes over here. You can't exclude this evolutionary descendant right here and say that this is a part of a clade. So that's why these are not clades. That's why these are not examples of clades. So, a clade has a root. This is the ancestral organism. And at each point when something happened that caused a divergence in which one characteristic became successful and leads to another species, and another characteristic becomes successful and leads to another species. Now, that node represents an ancestor. So this is an ancestor, and this particular, this point, this break in divergence is referred to as a speciation event. So something happened in nature that caused one characteristic to be more successful and another characteristic to be more successful in a different case. The fewer the number of nodes in between two species, like between A and B here, there's only one node, the more closely related these things are. Between A, B, and C, there's two nodes. So while C is related to this particular, like A and B, it's further apart from them than A and B are together. So we've got this ancestral species right here, and at that speciation event, whatever that event was, I mean, it might be, an earthquake, it might be a drought, it might just be that, one group of organisms moved to or migrated to another part of a particular ecosystem. But that event caused one set of characteristics to be more successful than another set. And as a result, the frequencies of alleles changed and led to a new species. So here we have another ancestor, and then this ancestor had the same thing happen. There was some event that caused one characteristic to be more successful for A and a different characteristic to become more successful for B. And now you have different species. So, how are these cladograms determined? Well, first of all, they can be determined by using structural evidence. So structural evidence is when scientists look at, you guys ready for a big word here? Scientists look at morphological differences. So morphology refers to body shape. So morphological differences will tell you when things became different from one another. So if a species is closely related, it should have more similar structures than things that are more farther apart. Okay. Now, it's an important thing to understand that we have been talking about evolution with respect to, differences that arise in things. So, for example, there are vertebrates that have a vertebral column, like a spinal cord, a cranium, a brain of different parts, and then those branched off into mammals, and then even amongst mammals, we had a lot of different speciation events that led to all of these different kinds of mammals. This is called divergent evolution when we have a branching like this. Now, when you consider structural evidence, it's important not to look at two things just on the surface. Because if you look at sharks and dolphins, you might say that they're closely related, but we know they're not. Dolphins are mammals. They give live birth. They nurse their young. They have lungs. Their skeletons are made of bones. But sharks don't breathe with lungs. They breathe with gills. Sharks don't have a skeleton made of bone. Theirs is made of cartilage. Dolphins don't have scales. Sharks have scales, and a very unique kind of scale. And so it's important to remember that when we're looking at physical characteristics, not just to focus on one, but to look at a few. So the fact that sharks and dolphins are very similar is a kind of evolution called convergent evolution, which means that they are sharing a certain selection pressure. Now, in this case, the pressure, the thing that nature was saying, like, hey, you've got to be able to do this in order to survive here, is being able to move well in water. And it just so happens that there's not very many ways to move well in water. And the best way is to be streamlined, to have certain fins in certain places, and so on and so forth. This is called convergent evolution. And this is not an example of common ancestry. So this is not an example of common ancestry. And so when you're making a cladogram, you can't just use one characteristic. You need to go a little bit deeper, and you need to look at different things just to make a better decision about what kinds of things are related to each other and what kinds of things aren't. And this brings us, as a refresher, do you remember when we talked about homologous structures as evidence for evolution? Homologous structures have the same structure and different function. And this shows that evolution can take place because this is losing my train of thought here. Same structure, different function. This is showing the adaptation feature of evolution. All right. Analogous structures are structures that have the same function, but they have a different structure. And because they have a different structure, it doesn't show a common ancestry. So analogous structures cannot be used in determining relationships in a cladogram. They cannot be used. So finally, last thing is, another piece of evidence that can be used to make a cladogram is molecular evidence. So what we can do is we can look at the structure of certain molecules and we can make a determination about how things are related by how similar these molecules are to each other. So since all organisms use DNA and RNA as hereditary pieces of information, since we all use them in the same way, then these DNA and RNA molecules can be used to determine how similar things are. Because remember that characteristics change because of mutations in the DNA. So, and over time, these mutations build up. They accumulate in our DNA. So the more similar we are to something, we could assume that we have fewer mutations in our DNA when you compare us with that thing. So we can compare the number of bases or the kinds of bases in our DNA strands and make a decision about these things. So when it comes to DNA, there's three different parts of the DNA that scientists can use. Let's start with number one here. Scientists can use non-coding DNA. Remember that not all of your DNA is used as blueprints to make proteins. In fact, it's a very small portion of the DNA that is used to make proteins. Okay, so the non-coding DNA is the best. This is the best to use to determine sequences because mutations occur more readily here. And what I mean by that, I'll make clear in the next step. So number two, we could be using gene sequences, which are the, the coding portions of DNA. These mutate at a slower rate. And the reason they mutate at a slower rate is because these mutations could affect protein structure, and it could be a change so dangerous that that particular organism doesn't survive. And so you can't have a lot of changes to protein structure, and that's why these mutations occur at a slower rate in these gene sequences, which is why it's okay that these mutations accumulate in non-coding DNA because it's not affecting protein structure. Okay, so finally, and this is not DNA, but it's close to DNA, and we use DNA to make proteins. Remember, proteins are made of amino acids, so we can compare amino acid sequences between similar proteins to determine how closely related things are between similar proteins. Now, these have the slowest rate of change because of something called codon degeneracy. And so remember when we looked at a codon chart, and I don't have a codon chart right in front of me, but we'll remember this, that if you had like AAA and AAU and AAG, all of those might have been the code for, amino acid 1. And then AAC would be amino acid, 2. Well, we wouldn't see a mutation in an amino acid if we were only looking at amino acids, and if one codon said AAA and one said AAU, because they're coding for the same amino acid, we wouldn't see that mutation until we looked at the DNA strand. So these amino acid sequences are normally used to compare really distantly related species because we can't see these mutations right here. So, for example, let's look at the hemoglobin. Remember, hemoglobin is made of 4 parts, and it is 2 alpha parts and 2 beta parts. So this is just looking at the beta part of hemoglobin right here. It's 146 amino acids in humans, and when you compare our amino acid sequences to a rhesus monkey, now rhesus monkeys aren't apes, they're monkeys, they have tails. And so they're a little further away from us than apes are. You'll see that there are only 8 amino acids that are different between us and a rhesus monkey. In a dog, there's 32 differences. We share 114 amino acids with them in this particular protein chain. Birds, we share 101, have 45 differences. In fish, we only share 21, 125 differences. And so if you were to make a cladogram based on this, you would say that humans and rhesus monkeys are the most closely related. So that would be us here. So this is human, this is a rhesus monkey. Dogs are a little further away from us. Birds are a little further away from that. Fish are even further away from that. And we have some common ancestor all together because there are some similarities there. So, tomorrow you are going to learn how to use actual DNA sequences that you can obtain from the internet to try to figure out similarities and differences between structures, between organisms, and make a cladogram based on that. All right, that's it for today.